thank you very much. Thank you very much for such a kind introduction. And also I would like to thank Spela and organizer for inviting me and my colleagues to wonderful city of Ljubljana. Thank you very much for that. And now I will try to actually describe our part in this fascinating project. So I'm coming from Czech Center for Phenogenomics, which is large research infrastructure in almost Prague, let's say few meters from the Prague border, from some funding reasons. And what we are actually doing, we are as a research infrastructure focused on the work with the mouse models from actually level of the genome editing to characterization of mouse phenotypes and also to preclinical development. As I said, we are center just next to Prague, and you can see that we are not so small team. We have 150 people roughly, and actually 10,000 square meters place where we do research and keep mice. And what to imagine when we are talking about research infrastructure. So we are not company, but we are also not the academic lab. So you can imagine it like these wheels, and that we actually have our own research program, but we also provide the service, assistance, and everything is driven by capacity and the philosophy of open access, which is actually very smart philosophy, which actually was seeded by European Union to research infrastructures which were built throughout Europe, that basically anybody can ask for our capacity, and we are supposed to help or handle the project if our capacity allows. And this is what actually happens with CTNNB1 project. And that's why I'm also here. So we are not the local infrastructure. We are part of the infra frontier. So you might know the infra frontier is the European level infrastructure connecting all, let's say, mouse centers over the Europe. We are also part of the global consortium IMPC, which is working for full annotation of mouse genome. And also we are part of EuroPDX consortium, which is focused on humanized cancer models for preclinical research. To achieve that, actually my team is called Phenotyping Module, and we are distributed in a couple of units which are focused on different parts of physiological investigation. So when we have a mouse model, we are able to handle it from the clinical biochemistry to behavior analysis and everything in the middle. We have also a team for working with the infection models and the cancer models. And what's actually our, let's say, direction where we are going is to move from the mouse genomics to understanding of human genes. So we are using our transgenic technologies to also design and prepare we call it avatar models, where we introduce the exact genetic mutation from patients into mouse model, and then we can characterize and also design therapy or eventually use it for preclinical development. So, and to achieve that, we have highly specialized preclinical pipelines where we are trying to use maximum modalities which we develop on a background of comprehensive phenotyping for mouse genome annotation, and we are using that for testing of new drugs, but also drug repurposing projects. And last 2 or 3 years, we are also developing the gene therapy programs for that. And this is our local activity in the Czech Republic. So we actually are working in this consortium of, I would say, the most significant institutes in Czech Republic in the development of gene therapy technologies. So for my team is focused mostly on neurodegenerative diseases as Angelman syndrome, Prader-Willi, and skin-related diseases. But other partners are also following different types of rare diseases and we are trying to share our technologies and approaches how to. So, but now let's go to really hard reality, and that's what we need for this project, and that's to run the GLP based study for toxicity. And it was just introduced before I came here what the GLP is, so I will go quickly through. We can actually conclude here that it's just the standard which unifies the laboratory approaches and makes them comparable and reproducible actually between countries, between partners. Where does it apply? So you can see it's not just only for the therapy, but also for the cosmetics, veterinary, food, feed, and also industrial chemicals. So the GLP is basically almost everywhere where you need to get the product to a human or in a chain which is leading to human consumption. So why is that? And this is very important because you can imagine just if you heard the example that it's not pretty difficult to do a tox study in a lab. You don't need much, and basically it's quite straightforward procedure. But the GLP is here that the quality of tests is as high as possible, that the data are reliable, and very important specifically from the medical development is the reproducibility of data and also the comparability of data. This is extremely important when you imagine to develop such a global product like, for example, gene therapies. But what it means really from the perspective of somebody who has team who is supposed to work in a GLP. So you can imagine from this cartoon says basically everything. You can imagine that every part which is included in GLP, which means the facility, organization, all personnel, all devices, all reagencies, all material, all protocols, everything has its own paperwork. So, for example, recently we already reached the number of 180 SOPs which are signed by authority and are regularly audited. And with this study and other studies coming, we will break 200 very soon. And this is something what you keep paperworking and something what could be done with one person, you realize that you need at least 5. And and this is basically never-ending story. But what is important, there is a need for that, and there is a good reason to have it. And that's why we are doing that. But what is extremely important, you can imagine the tons of papers which are behind. But what is very important is that every study must have very detailed study plan. This is critical. This is not just like waking up and saying, okay, today I will test whether it's toxic or not. You have to prepare the study plan in advance. And this study plan must be actually authorized by the, let's say, our National Drug Control Authority, and they can put some additional readouts or tests which they want to have in a tox study. So this is something also what we will need to clarify for beta-catenin study, but basically I think we are in a really good way to achieve this. So we need the consultation with the authority, and the authority confirms that the study plan is according the best of their practice. And now we are getting to a study which will be housed in our center. So here are the details of the studies. So we will have 2 cohorts. One will be short, one will be a bit longer. And maybe I can skip the boring text and go to cartoon. So so we will use GLP validated ICV injection in a stereotactic frame, where we basically are using the state-of-art technology to deliver the small volumes into brain ventricles. And then actually we will wait. We will weight animals, we will check their vision, and in a terminal phase, we will collect the blood samples, urine samples, and we will do detailed necropsy. So specifically brain, but all organs will be taken by 2 trained and audited pathologists, which will provide provide the detailed histopathological analysis of all organs which are actually in a mouse, actually. And the important is that there will be actually pathological evaluation of all possible changes. We should be prepared that there might be some background lesions also in a wild type animals, because we are working with the live natural system where is also certain level of randomness. So it can happen that, for example, wild control mouse can have a lesion in a gut, or for example, stone in kidney or something like that. And all of this must be reported and evaluated whether it's relevant to treatment or not. So this is probably the most tricky part, to have the proper histopathological analysis, but otherwise you can see that it's pretty straightforward design. The little tricky is the injection into brain, and the rest is very simple experiment. But just imagine how many papers must be signed to actually finalize this study. But we will make it. And here I would thank you for your attention, and I'm really eager to take any question to this problematic, if you have any.
You will get questions in the round table.
Okay.
Yes.
So I'm leaving.
Yes.
Thank you.